Hello, and welcome to our webinar, How to Optimize Non-Viral CRISPR HDR for High-Efficiency Large Knock-in in Primary T Cells and iPSCs. My name is Tyler Kozushek, and I'm a Senior Scientist at Aldevron, and will be serving as the moderator for today's presentation. I have a PhD in Biomedical Engineering from the University of Nebraska-Lincoln, where I studied non-viral gene delivery to primary human mesenchymal stem cells. I currently lead the nanoplasmid design team at Aldevron. Today's webinar will be presented by Bernice Thermandrew. Bernice is a research scientist in the Molecular Genetics Research Group at IDT. She received her master's in molecular physiology and biophysics from the University of Iowa, where she studied transcriptional regulation of multi-drug resistance in pathogenic fungi of the lung. At IDT, Bernice has focused on optimizing delivery strategies for CRISPR reagents, as well as methods for increasing the rate of homology-directed repair. During today's webinar, please submit your questions using the question box. Any questions that are not answered during the Q&A session will be followed up afterwards. And with that, we'll take it to you, Bernice.
Thank you very much, Tyler, for the introduction. Hi, everyone. Welcome to our webinar titled How to Optimize Non-Viral CRISPR HDR for High-Efficiency Knock-in in Primary T Cells and iPSCs. Again, my name is Bernice Thermandrew. I'm an R&D scientist at IDT, and together with my colleague Tyler from Aldevron, today we're going to present about 3 years worth of data and optimization work that we've done at IDT to improve our CRISPR editing and HDR levels in clinically relevant cells. So let's jump into it. So here's an overview of what we'll talk about today. We'll start with a bit of background on basic principles of CRISPR editing. Then we'll discuss the proper cell handling workflow for CRISPR editing in primary T cells specifically. Then we'll show some optimization data for use of Aldevron's nanoplasmids for both iPSCs and primary T cells. We'll wrap up with some conclusions, and then we'll end with answering questions from the audience. So please do feel free to submit questions as we go along. We'll answer as many as we can at the end of today's presentation. So let's talk about basic principles of CRISPR. And CRISPR's been around for about a decade. I'm sure plenty of people know at least a little bit about it, but I want to point out some key components here, starting with the idea that CRISPR at its core is a method to target genomic DNA. So with a CRISPR-Cas9 nuclease, there's a targeted intended double-strand break. And the interesting part of CRISPR really comes in the repair of that double-strand break. And that's where different reagents can impact the repair outcome. So following a double-strand break, there are a few different repair pathways that the cell can use for repair. And non-homologous end joining, or NHEJ, is most common for making a gene knockout because it gives rise to random indels. Mainly today, I'll talk about another pathway called homology-directed repair, or HDR, which is when a sister chromatid, or alternatively, an exogenous donor to repair template can be provided that the repair machinery uses to repair that double-strand break site and can bring a desired insert along with it. For some background on CRISPR HDR, here's a cartoon graphic outlining the steps that one would take to create a knock-in. So you'd start with designing an HDR donor template, which contains sequence that immediately flanks the guide RNA cleavage site on either side with your desired insert in the middle. And there are lots of options for donor template formats like viral vectors, double-stranded DNA, single-stranded, plasmid, mini-circles, etc. Today I'll mostly focus on nanoplasmids, so that's a long, double-stranded circular template. And next you would want to deliver your CRISPR reagents to generate that targeted double-strand break and allow the HDR donor to homologously recombine into the genome and bring the insert with it. And the last step is analysis, which can be done by sequencing. We tend to use a lot of sequencing like Illumina or Oxford Nanopore, or by phenotyping like flow cytometry or ELISA. And for small inserts, like inserting a stop codon or making a SNP change, or even a small epitope tag, we recommend using single-stranded donor oligos like IDT's Alt-R donor oligos, which is made through our ultramer chemistry and has some additional chemical modifications on the 3' and 5' ends. You can find lots of information on those on our HDR methods paper in scientific reports and also on our website. And all of the data that went into this optimization also went into make IDT's HDR design tool. And this is a free tool on our website that can aid in guide RNA selection and donor design. So check that out for all of your short insert needs. So now I want to transition into talking about what's specific about HDR for primary T cells. So as many of you have probably heard, these cells can be tricky to work with and even more tricky to do large knock-ins with. Much more difficult than a standard immortalized cell line or a stem cell like iPSCs. And one significant factor of T cell editing is the donor of the cells themselves. So here in this graph, we've delivered RNP to 7 different genomic loci in T cells isolated from the blood of 3 unique human donors. And this was a direct comparison in the same experiment. So the editing levels we observed here are consistent with what we've seen many times. The trends of editing, like what guide RNA is optimal, will stay the same, but the baseline and the potential of editing can vary from donor to donor. And because of that variability, we always recommend testing multiple donors side by side for true biological replicates to understand where the baseline for editing is for each donor. And as with many cell types, we found that the key to efficient CRISPR editing and HDR in primary T is to find the balance of where you have enough donor template to achieve high HDR rates, but not so much that the health of the cells is negatively affected. So we also recommend doing dose titrations of at least just the donor template. So here on the x-axis, we have a dose range of 0 to 10 micromolar. Our standard recommendation is about 3 to 5 micromolar. And you can see in the blue line, about 5 micromolar is where you see the optimal HDR. But also consider any additional reagents that are added, like the electroporation enhancer that we used here, and that's shown in the red line. And adding that reagent changes the optimal dose of the donor, where it ends up around 3 micromolar. And here in IDT R&D, we continue to research and optimize all donor template modalities in as many different use cases as we can. And for a large knock-in, we identified Aldevron's nanoplasmid vector system as a very efficient tool for knock-ins that are over 200 base pair. So the small backbone, high yield, and low toxicity are ideal for HDR in primary T cells where the cells are a bit more sensitive. And we consistently see high HDR rates when using nanoplasmids, as you'll see over the next few slides. This slide shows a comparison of template modalities in primary T. So this is 2 different targets and 2 different insert sizes. On the left is a GFP knock-in, so that's about 800 base pair. And then on the right is an anti-CD19 CAR knock-in template, so that's about 2 kb. And it was in this type of comparison that we really observed that nanoplasmid is a great choice for large knock-in in primary T. And what's labeled as competitors here, this is a mix of linear double strand, linear single strand, circular single strand, always comparing back to nanoplasmid as our internal control in the blue bar. So for these 2 targets and these 2 different insert sizes, nanoplasmid gave us the highest rate of HDR, while some templates really didn't even reach moderate levels of efficiency. And now I want to talk about the cell handling workflow for primary T cells. Primary T have very specific needs as far as cell handling. And so in the graph on the left, we tested some different commercial T cell activators for their effect on HDR rate. So these are commercially available T cell activators, starting with Immunocolt from Stem Cell Technologies, Dynabeads from Thermo Fisher, and Transact from Miltenyi. And we found some trends here using both Cas9 RNP and Cas9 mRNA. The highest HDR rates that we saw were always with Miltenyi, Transact. And then on the right, we experimented with different time points of activation using that Transact. And we did that and measured CAR knock-in rates. So the 4 conditions we tested were no activation, pre-activated for 48 hours, which is the manufacturer's directions and what is kind of standard for the field. Then no pre-activation, but activated immediately post-transfection. Then both pre-activated for 48 hours and reactivated immediately post-transfection. And again here we saw some clear trends that with no pre-activation at all, then no HDR occurs. And that kind of makes sense considering the complex repair machinery that needs to be recruited for HDR to occur. And if the cells aren't metabolically active, then that process just won't happen. And the standard method of 48-hour activation starts to show some moderate levels of HDR. But the highest rate of knock-in was observed when the cells are primed with the activation prior to transfection and then reactivated immediately post-transfection. And we think this is just because the cells are most metabolically active with that first activation and then the second. And this is now our standard activation protocol for any primary T HDR experiment. And another note on reagents here, you may have noticed that in some graphs that I'm showing, we used Cas9 RNP and some we use Cas9 mRNA. And there are different modalities of delivering Cas9 nuclease itself. You could do that as plasmid, as mRNA, as recombinant protein in the form of RNP, and all of these are shown in the figure on the left. But there are certain reasons why you would want to use one over the other. And for standard NHEJ or small HDR, like insertion of a restriction site or a SNP change, RNP is sufficient, and its short half-life in the cell helps prevent some off-target editing that could occur. On the other hand, the graph on the right is a direct comparison of Cas9 mRNA and Cas9 RNPs. And we see that for large knock-ins like GFP, or again that same anti-CD19 CAR insert, we do see a benefit of the longer expressing Cas9 mRNA to give the cell a prolonged chance of cleaving the target and inserting that larger sequence. Another important factor is the delivery method. At IDT, we typically use the Lonza nucleofection system. We see good efficiency with this and good cell viability, but the optimal delivery strategy can come down to the granularity of exactly what electroporation settings to use. And this graph is a comparison of Lonza nucleofection protocols, including some that were recommended by Lonza and some that were just identified from the literature. These were tested for their effect on HDR in primary T cells using that same CAR knock-in nanoplasmid template. And there are some clear differences in editing efficiency depending on the Lonza nucleofection protocol. So this is another step that can be optimized for everyone's specific use case. So when using Cas9 mRNA and nanoplasmid HDR templates, we use the optimal electroporation settings that we determined empirically. We talked earlier about how important the health of primary T cells is to the efficiency of HDR. So because of that, we knew that the dose of every reagent should be tested for the threshold of high efficiency and low toxicity. So here we test the nanoplasmid dose to ensure maximum HDR is reached and identified that 2 microgram is the lowest dose that leads to high HDR efficiency. So this is another reminder that more is not better. You can't just increase the donor template and expect HDR to increase as well. There is a threshold of where you'll see diminishing returns. I mentioned that the interesting part of CRISPR is how the cell repairs a double-strand break, and that there are reagents that can influence the repair machinery that the cell uses, and that can be true for either NHEJ or HDR. And at IDT, we have a couple of products that do exactly that. They influence the repair outcome. Our newest product is the HDR enhancer protein. So this product is coming soon, and its mechanism is to inhibit I53, which is an inhibitor of HDR. So you inhibit an inhibitor of HDR and increase the HDR rate without impacting NHEJ at all. And this can be used alone or combined with the HDR enhancer V2, that one is a small molecule, to boost HDR rates of small and large knock-in. Here, the HDR enhancer protein is used to increase the rate of HDR of a CAR knock-in using Cas9 mRNA, IDT sgRNA, and the CAR nanoplasmid at 2 different loci in the TRAC gene. So here we see a site-dependent effect on the fold change observed with the enhancer, and that can be true for varied sites depending on the specific sequence context of your target and the insert. And here at IDT, we're always looking into clinically relevant cells, and I want to mention that nanoplasmids work great in iPSCs too. So again, here we're using Cas9 mRNA to mediate a double-strand break. We're using sgRNA and a nanoplasmid insert template. On the left is a comparison of homology arm lengths, and the right is a comparison of insert sizes. So in all these cases, high rates of HDR are achieved with careful optimization, with use of the HDR enhancer protein and use of nanoplasmids, so we can do high efficiency knock-in in both primary T and iPSCs. And so this is the full workflow that I spent 3 years optimizing, so you don't have to. So we perfected specifically this workflow for large insert HDR. The IDT guide RNA design tool can help identify guide RNA in your target region, and the nanoplasmid team at Aldevron can help design a DNA template with your desired insert. So when T cells are thawed, we use a specific activator and IL-2 at a high dose. The cells are incubated for 2 days and then transfected with CRISPR reagents. So what we've always seen the best results with is Cas9 mRNA, single guide RNA, and a nanoplasmid HDR template. And then the activator is included in the post-transfection media. So another 2 days of incubation, and then typically the cells are ready to be analyzed, but this depends again on your assessment method and the expression of your desired transgene. And finally, if you're analyzing by NGS, then the RAMP-seq CRISPR analysis system can help you through analysis and histogram generation. So let's summarize with some conclusions here. Today's presentation discussed how to improve knock-in rates in primary T cells using specific reagents like nanoplasmids and HDR enhancer protein. And we know that primary T cells can require a bit of scrutiny to perfect the reagents and workflow to achieve the desired editing outcome. And nanoplasmids are a great choice for large knock-in, considering the small backbone prevents high DNA toxicity, and they can be manufactured in high yields. And finally, you can use reagents like the HDR enhancer protein to modulate double-strand break repair pathways and improve the rate of HDR. And last thing I'll mention here, IDT has been your trusted partner for RUO reagents for a long time, and our goal is to support our customers through every stage. So we built out our cGMP manufacturing capabilities to offer cGMP guide RNAs for therapeutic CRISPR editing applications. So whether you need small-scale batches for preclinical or large-scale production for clinical trials, we offer flexible manufacturing solutions that can suit your project's needs. So now the same care and attention that IDT has always paid to our RUO customers, we want to provide with a full workflow for every stage through RUO, engineering run, and cGMP. And we can help at every stage that goes along here. So project consultation, custom manufacturing solutions, QA, and regulatory compliance. We're here to help. Alrighty, that's it for today. Thank you both to my team and to Tyler's team at Aldevron. Tyler, we're happy to take it away with any other questions from the audience. Thanks everyone for listening today, and submit your questions in the chat and we'll get to as many as we can.
Thank you, Bernice, for a very informative presentation. We have received several questions from our audience. The first question is, why does the activation state of T cells matter for CRISPR efficiency?
So there's an important distinction to make here. It doesn't matter as much for NHEJ if you just need to make a knockout, because the Cas9 will get in and cleave DNA no matter what the state of the cell. It definitely matters for HDR because that's more of a energetically demanding repair process. So the cells need to be metabolically active in order for HDR to occur. So that's why we have to time the activation in a particular way to increase the efficiency of HDR.
Thank you, Bernice. Another question here. How do you measure cell viability and HDR efficiency?
For cell viability, we do flow cytometry or occasionally the Presto Blue assay, which is a commercially available Resizurin conversion reagent for measuring cell health. For measuring HDR efficiency, we would again use flow cytometry or occasionally NGS in the form of Illumina sequencing, and also long read ONT sequencing.
Perfect. Yeah, some good questions here. Please keep these coming. How much variability is there in editing efficiency between different cell donors?
Great question. A lot. There's a lot of difference between different donors, just like every human being is different. It can be due to a number of factors like the chromatin state or any specific SNPs that a person would have. We don't necessarily see differences in trends for different reagents or doses, but generally the baseline and the potential for CRISPR editing is what varies donor to donor.
Okay. And when should I use a nanoplasmid instead of an SSODN?
Great question. IDT can make SSODNs up to 200 nucleotides. So that's our ultramer chemistry and our HDR donor oligos. So up to 200 nucleotides for that, and anything larger, we would go with a donor block or a nanoplasmid template.
We've got a few more questions here. How do you use the HDR enhancer protein?
Yeah, it's pretty easy to use. It just gets added along with all the other CRISPR cargo in your nucleophexin reaction or whatever delivery method you use. So along with RNP or Cas9 mRNA and your donor template, the HDR enhancer protein is a liquid reagent that just gets spiked into that reaction and delivered along with everything else.
All right. And when will the HDR enhancer protein be available for purchase?
Great question again. Early access for the RUO protein will be available mid-next year. So definitely reach out to us if you want to get on the early access list. And then GMP will come along shortly after that. So we'll have both an RUO and GMP version of the HDR enhancer protein in the next year.
We got time here for one last question. How do I order a custom nanoplasmid for HDR studies? I can take this one here, Bernice. The ordering process for custom nanoplasmids is relatively simple. You can send your molecule files to nanoplasmids@aldevron.com. These molecule files can be a puck vector that you are currently working with, a custom insert that requires de novo synthesis, or a combination of the two. Our nanoplasmid design team will look over the molecule files that you sent, design the nanoplasmid versions, and send these designs back to you for review. Once the final design is approved, clients then can receive a formal quote for the order from Aldevron. All right, well, unfortunately, we have run out of time to answer any additional questions. We will contact you directly if you asked a question and it was not answered here today. I want to thank you again for attending, and we wish you the best of success in your research.
Thanks, everyone.